Okay, let's dive in. Seriously, think for just a moment about something you do every single day, maybe without giving it a second thought, like reaching out and picking up a glass of water, or even simpler, just staying upright in your chair right now without tumbling over.
Right. Seems effortless.
It feels completely automatic, doesn't it? Like second nature. But what if I told you that beneath that seemingly effortless action lies an absolutely mind-blowing, incredibly complex orchestration of signals and systems crisscrossing your brain and spinal cord?
It really is a true feat of biological engineering. What presents as simple, fluid movement or stable balance is actually the result of lightning-fast neural communication, constant adjustments, and complex feedback loops happening way below your conscious awareness.
Exactly. And that hidden complexity is precisely what we're here to unpack today. This deep dive is all about pulling back the curtain on the incredible neural machinery that makes voluntary movement and equilibrium possible.
Yeah, we've got a stack of detailed source material in front of us. Articles, research findings, deep dives into the anatomy and physiology, and we're going to try and navigate it together.
Our mission with this material is really to guide you through the different layers of your brain and spinal cord involved, help understand how they collaborate, and truly uncover the surprising sophistication behind even the most basic physical tasks.
Think of this as your shortcut to understanding the inner workings of movement and balance. We're really aiming to grasp this material at a level of approaching, mastery.
So settle in, trusting your brainstem to keep you there, and let's start our exploration at what's often considered the command center for action, the motor cortex.
Okay. So when we talk about initiating a movement, like deciding to pick up that glass, the signal often originates in the cerebral cortex.
The thinking part.
But here's a crucial insight right away from our sources. These voluntary movements don't happen in a vacuum. They rely heavily on established patterns of function already residing in lower brain areas. That's the spinal cord, the brain stem, and deeper structures like the basal ganglia and the cerebellum.
So it's not like the cortex is inventing the movement from scratch every single time. It's more like it's sending a signal to activate a preloaded program stored somewhere else.
That's a really good way to picture it. Yeah, it's activating a highly refined subroutine. However, the sources make an important distinction.
Okay.
For some movements, particularly those requiring very fine, precise control, think threading a needle or, playing a musical instrument with your fingers.
Uh-huh, the really dexterous stuff.
Exactly. The cortex does have a more direct line. This direct pathway bypasses some of those lower pattern generators, which highlights the cortex's specialized role in dexterity.
Okay, let's ground this in anatomy. Where exactly is this motor cortex we're focusing on?
Well, looking at the diagrams provided, the primary motor cortex is located just in front of a major groove on the brain surface called the central cortical sulcus.
Okay.
It sits in the posterior part, the back third of your frontal lobes.
And that central sulcus is also the boundary with the somatosensory cortex, right? The area that processes touch and body position.
Absolutely. And this geographical closeness is no accident. The sources emphasize that the somatosensory cortex, which tells you about what you're touching, where your limbs are, how they're positioned.
All that incoming info.
It feeds a wealth of information to the motor cortex. You need this sensory context to initiate and refine appropriate movements. You need to know where your hand is before you can plan to move it effectively.
So sensory input isn't just passive information. It's actively involved in the motor command itself. It's part of the planning.
Precisely. Now, the motor cortex itself isn't just one uniform area. Our source material breaks it down into 3 distinct sub-areas, each with its own map of the body and its own specialized role in generating movement.
Okay, what are they?
These are the primary motor cortex, the premotor area, and the supplementary motor area.
Let's start with the star of the show, the primary motor cortex.
Right. This area, also known as Brodmann's Area 4, is located right on the first prominent ridge of the frontal lobe, just anterior to that central sulcus.
Okay.
It wraps around the side of the brain near the sylvian fissure and curls up over the top, even dipping down into the deep groove between the hemispheres, the longitudinal fissure. The figures in the source material really show this mapping beautifully.
And the map is, well, it's not what you might expect, is it? It's not a mini you laid out neatly.
No, not at all. It's strikingly distorted. This mapping was famously explored by neurosurgeons like Penfield and Rasmussen back in the mid-20th century.
How did they do that?
They would electrically stimulate points on the exposed motor cortex during surgery unconscious patients
Wow.
and observe which muscles twitched.
Talk about a unique patient experience.
Right. And what they discovered was this homunculus, this distorted representation of the body. The face and mouth area is represented laterally near the sylvian fissure. The arm and hand are in the middle portion. The trunk is near the top, and the legs and feet are tucked inside that longitudinal fissure.
And the disproportionate size of certain areas is a key insight, isn't it?
Absolutely crucial. The sources highlight that more than half of the entire primary motor cortex surface is dedicated to controlling the muscles of the hand, the fingers, thumb, palm, and the muscles involved in speech. The mouth, tongue, larynx.
More than half. Just for hands and speech.
Yep. This tells you immediately how incredibly important fine manual manipulation and complex communication are to human function. The amount of brain real estate directly reflects the complexity and importance of the movement.
Okay, that makes sense.
Precisely. Another vital finding from those stimulation studies, noted in the source, is that stimulating a single point in the primary motor cortex rarely causes just one muscle to contract.
Really? So it's not one spot, one muscle.
More often, it activates a group of synergistic muscles working together. This led to the understanding that a single neuron here doesn't just control a single muscle fiber. It contributes to activating a pattern of muscles needed for a specific movement.
Like activating the pattern for reach and grasp rather than just, say, firing your bicep in isolation.
Exactly. That pattern involves various muscles coordinating their contractions with the right direction and strength to achieve the intended action, like reaching for that coffee cup.
Okay. Now, moving just forward of the primary motor cortex, we find the premotor area.
Right. Located 1 to 3 centimeters anterior to the primary motor cortex, the premotor area also extends laterally and superiorly, bordering the supplementary motor area. It has a rough topographical map, somewhat similar to the primary motor cortex.
What's its main job? The source suggests it generates much more complex patterns of movement.
Yes, exactly. While the primary motor cortex handles the individual, sort of, discrete movements, the premotor area is more like the choreographer. It's involved in planning and sequencing more elaborate movement sequences.
Choreographer, I like that.
It's particularly important for positioning the body or limbs in anticipation of a movement, like preparing your shoulder and arm posture before you even start to reach out.
The source mentions a two-step process involving a motor image. What's that about?
This is a helpful concept for understanding its role. The anterior part of the premotor area seems to develop a kind of mental blueprint or motor image of the entire movement sequence you're about to perform.
Like planning the whole thing out first?
Kind of. Then, the posterior part of the premotor cortex takes that image and excites the successive patterns of muscle activity in the primary motor cortex and other areas needed to execute that complex sequence.
And the pathways from the premotor area are interesting. They don't always go straight down to the spinal cord, right?
That's right. While some signals do go directly to the primary motor cortex to trigger specific muscle groups, a significant portion of premotor output travels a more indirect route. They often pass through the basal ganglia and the thalamus, looping back to influence the primary motor cortex.
Just leaping back.
Yeah, this emphasizes the deep interconnectedness with these subcortical structures involved in motor control. It's not just a top-down command.
And this is also the location of those fascinating mirror neurons we hear about.
This is where we find them. According to the source, a special class of neurons in the premotor area that are active both when you perform a specific motor task and when you observe someone else performing the same task.
So watching me reach for my water might cause some of the same neurons to fire in your brain as if you were reaching for it yourself. That's wild.
Exactly. Their activity literally mirrors the behavior you're watching. Brain imaging studies cited in the source support the idea that these neurons transform sensory input, whether you see or hear an action, into a motor representation.
What's the thinking on why we have them?
Well, many neurophysiologists believe they are fundamental for understanding the actions of others and for learning new skills through imitation. It's a fascinating link between perception and potential action.
So the premotor cortex, working with the basal ganglia, thalamus, and primary motor cortex, is essentially the conductor for complex, coordinated muscle activity.
It's key in translating a general intention into a detailed, sequenced motor plan.
Okay, the third subarea is the supplementary motor area. Where's that and what does it do?
It's found mainly within the longitudinal fissure, that deep groove between the hemispheres extending onto the superior surface of the frontal lobe. It also has a topographical organization for motor control.
What distinguishes its function from the premotor area?
The sources note that stimulation of this area often results in bilateral contractions, movements on both sides of the body simultaneously.
Both sides at once, like what?
Think of grasping with both hands, perhaps related to primitive actions like climbing, as this source suggests, or maybe stabilizing the body.
And it works closely with the premotor area.
Absolutely. While the premotor area focuses on sequencing specific actions, the supplementary motor area provides the broader context and background. It's involved in body-wide attitudinal movements, fixing certain parts of the body like stabilizing your trunk or shoulders while other parts move, and controlling positional movements of the head and eyes.
Ah, okay. So it provides the stable platform.
Exactly. This provides the necessary stable platform upon which the finer movements controlled by the premotor and primary motor cortex can be executed. It provides that foundational, often bilateral, postural support.
In addition to these three main areas, the human cortex also has some highly specialized regions for motor control, right? Identified by what happens when they're damaged or stimulated.
Yes, these are critical for very specific functions. One of the most well-known is Broca's area, or motor speech area.
Located in the premotor area, just anterior to the primary motor cortex for the mouth and tongue, and above the sylvian fissure. The source diagrams label it word formation.
Correct. Damage to Broca's area is quite specific. A person doesn't lose the ability to make sounds, but they lose the ability to form coherent words and sentences.
So they can make noise, but not meaningful speech.
Right. The source describes the output as uncoordinated utterances, or just occasional simple words. It's the loss of the complex motor program for speaking whole words.
And it's linked to breathing control for speech, too.
Yes, the sources mention an associated area that controls the precise respiratory function needed to coordinate breathing with mouth and tongue movements for speech. It really underscores the incredibly intricate neuronal activity required just to speak. It's amazing.
Then there's a specialized area for controlling voluntary eye movements.
Situated in the premotor area, just
Yeah.
above Broca's area. Damage here impairs the ability to move your eyes voluntarily towards objects. Instead, your eyes might involuntarily fixate on whatever the visual areas in your occipital cortex are focused on.
So you can't choose where to look easily.
Right. This area also controls eyelid movements, like blinking.
And slightly higher up is an area for head rotation.
Yeah, stimulation there causes head rotation, often directed towards objects. It's closely linked to the voluntary eye movement field, helping to orient the head and eyes together. Makes sense, right?
Yeah, coordinate looking and turning. And the area for hand skills.
This is in the premotor area, just anterior to the primary motor cortex region for the hands and fingers. It's crucial for learned, skillful hand movements.
Like playing an instrument or typing.
Exactly. The source notes that damage from tumors or lesions in this area can lead to motor apraxia, uncoordinated, non-purposeful hand movements. The person has the muscle strength, but has lost the complex motor programs for skilled use of the hand.
Okay, so we've covered the planning and specialized areas in the cortex. How do these commands get translated into signals sent down to the spinal cord and muscles? What are the pathways?
That's where the transmission pathways come in. The source describes two main routes from the motor cortex. The direct route, which is the corticospinal tract, and indirect routes involving accessory pathways that loop through the basal ganglia, cerebellum, and brainstem nuclei.
And the direct route, the corticospinal tract, is the main one for those fine, discrete movements we talked about.
Exactly. The source highlights that these direct pathways are particularly important for the discrete, detailed movements of the distal limbs, meaning the parts furthest from the body, like the hands and fingers. Again, this aligns perfectly with the large cortical representation of those areas.
Okay, let's trace that most important pathway, the corticospinal or pyramidal tract. Where do the fibers actually start?
Well, the sources give a surprising breakdown. About 30% come from the primary motor cortex.
Okay, expected that.
Another 30% from the premotor and supplementary motor areas.
Also makes sense.
And interestingly, 40% originate from the somatosensory areas just posterior to the central sulcus.
Wait, 40%?
Wow.
From the sensory side, that's huge. It really emphasizes that sensory information isn't just input before the command, it's part of the command pathway itself.
Precisely. That integration is absolutely key to precise movement. Once they leave the cortex, these fibers funnel down through a critical narrow passage called the posterior limb of the internal capsule. This is a common spot for stroke damage because so many important fibers pass through it.
A bottleneck.
Very much so. Then they continue down to the brainstem, where they form visible bundles on the front of the medulla, known as the pyramids. That's why it's also called the pyramidal tract.
And this is where most of them cross over to the other side.
Yes, a vast majority of the fibers cross over to the opposite side of the brainstem in the lower medulla. They then descend in the lateral corticospinal tracts in the side portions of the spinal cord.
And where do they actually end up in the spinal cord? Do they connect directly to the muscles?
Mostly not directly. Primarily, they terminate on interneurons located in the intermediate gray matter of the spinal cord.
Interneurons, so middlemen neurons.
Yeah, exactly. A smaller number terminate on sensory relay neurons in the dorsal horn, perhaps modulating incoming sensory signals related to the movement. And a very small number terminate directly on the anterior motor neurons, the final output neurons that actually cause muscles to contract.
So even the direct corticospinal pathway mostly talks to these interneurons first, not directly to the final motor neurons.
Correct, with a key exception we'll get back to. The sources also mention a small contingent of fibers that don't cross in the medulla.
Oh, where do they go?
These travel down on the same side in the ventral corticospinal tracts, located in the front part of the spinal cord. Many of these eventually cross over lower down, in the neck or upper chest.
What do those do?
These fibers are thought to control bilateral postural movements, possibly linking back to the supplementary motor cortex's function in coordinating both sides of the body.
The fibers themselves are also diverse, not all the same.
Yes, there's a fascinating mix. The most striking are the large, heavily myelinated fibers, about 16 micrometers in diameter, which conduct impulses incredibly fast.
The express lane.
Pretty much. These originate only from the giant pyramidal cells, also known as Betz cells, found exclusively in the primary motor cortex.
Betz cells are huge neurons, aren't they? I remember reading about them.
They're up to 60 micrometers in diameter, and their fibers are speed demons, transmitting impulses at about 70 meters per second, the fastest rate from brain to spinal cord. The source says there are around 34,000 of these Betz cell fibers in each tract.
But wait, you said the whole tract has over 1 million fibers, so these fast Betz fibers are a tiny minority.
A very small percentage. Betz fibers make up only about 3%. The remaining 97% are mainly smaller fibers, less than 4 micrometers.
What are the smaller ones doing then?
They're thought to transmit background tonic signals to the spinal cord motor areas, maybe maintaining a baseline readiness or tone.
So the big fast ones might be for those sharp, decisive commands, the go signal, while the smaller ones provide the continuous underlying neural hum needed for motor readiness or sustained action.
That seems to be the functional distinction implied by the sources. The Betz cell pathway is likely responsible for rapid initiation, while the smaller fibers contribute to sustained control and background tone.
Okay, the corticospinal tract is clearly the most important output pathway. But you said the motor cortex doesn't just send signals down this one road. It connects with a lot of other brain areas simultaneously.
This is a critical point for understanding the distributed nature of motor control. The motor cortex sends signals everywhere. It's not working in isolation.
Like where else?
Well, there are short branches from the Betz cells that loop back within the cortex itself, potentially inhibiting adjacent regions.
Why would it do that? Inhibit itself?
The idea is it might sharpen the focus of the excitatory signal, like noise cancellation, making the intended movement signal clearer by silencing nearby, potentially competing signals.
Like a spotlight. Okay.
Exactly. A large number of fibers also project to the caudate nucleus and putamen, which are key components of the basal ganglia.
Ah, the basal ganglia. We know they're involved in movement control too.
Crucially, pathways from these areas are vital for controlling body posture and background muscle tone, a clear link to the subsequent sections of our dive, talking about posture and balance.
So as the cortex plans a movement, it's simultaneously telling the basal ganglia to prepare the body's foundation for that movement, setting the stage.
Yes, it's a parallel process. Moderate numbers of fibers also descend to the red nuclei in the midbrain, which gives rise to the cerebrospinal tract, another important descending pathway we need to discuss.
Okay, red nucleus, cerebrospinal tract. Got it.
Signals also go to the reticular substance and vestibular nuclei in the brainstem, feeding into the reticulospinal and vestibulospinal tracts that descend to the cord and also connecting to the cerebellum.
That's a lot of communication with the brainstem and cerebellum. It's clearly not just cortex to spine.
And it doesn't stop there. The sources point out a tremendous number of fibers that synapse in the pontine nuclei in the pons. These then send signals via pontocerebellar fibers to the cerebellar hemispheres.
So a massive pathway to the cerebellum too.
Massive. Plus, collaterals also go to the inferior olivary nuclei, which relay signals via olivocerebellar fibers, also to the cerebellum.
So the summary of these other pathways is really important. The basal ganglia, brainstem, and cerebellum all receive strong signals from the corticospinal system every single time the cortex sends a signal for motor activity.
This is a major takeaway. It means voluntary movement is not just the cortex telling the muscles what to do. It's the cortex initiating a process that immediately involves and coordinates activity in these other major motor control centers. They're all working together in a highly integrated dynamic system.
Okay, that's the output side. But before the motor cortex sends signals down, it needs information, right? What are the incoming sensory fiber pathways feeding the motor cortex? How does it know what to do?
Absolutely. The motor cortex's activity is heavily influenced by sensory input, primarily from the somatosensory system, but also vision and hearing. The motor cortex, basal ganglia, and cerebellum process this sensory information to decide on the appropriate motor response.
What are the specific inputs? Where does the info come from?
Well, there are fibers connecting from adjacent cortical areas. From the somatosensory cortex right behind the central sulcus, from the frontal cortex in front of the motor area, and even from the visual and auditory cortices.
So it's getting updates from nearby brain regions.
Yeah, showing how sensory and association areas inform motor planning. There are also connections from the other side of the brain via the corpus callosum, connecting corresponding motor and sensory areas in the opposite hemisphere.
Keeping both sides in sync.
Right. And crucially, direct somatosensory fibers come from the ventrobasal complex of the thalamus. This is a key relay station that brings in tactile information, joint position, and muscle signals directly from the periphery of the body.
So the motor cortex gets direct, real-time information about where the body is and what it's touching.
Exactly. And significantly, tracts from the ventrolateral and ventroanterior nuclei of the thalamus bring signals from the cerebellum and the basal ganglia.
Ah, so those loops connect back up to the cortex too.
Yes. This is absolutely vital for the coordinated communication and interaction among the motor cortex, basal ganglia, and cerebellum. They are constantly informing each other's activity. It's a continuous dialogue.
And finally, fibers from the interlaminar nuclei of the thalamus. What do they do?
These project to the motor cortex and play a role in controlling its general level of excitability, kind of like adjusting the volume or sensitivity, influencing its readiness to respond to inputs.
So while the corticospinal tract is the main output highway, the motor cortex is clearly part of this massive interconnected network, constantly receiving information from sensory areas, association areas, and these other big motor control centers like the basal ganglia and cerebellum.
It's a complex loop. Definitely not a simple one-way street from command to action.
Okay. We talked about the corticospinal tract as the most important direct pathway, but the source also details an important alternative route involving the red nucleus. Tell us about that.
Right, the red nucleus. It's a structure located in the midbrain, or mesencephalon. The sources describe it as functioning in close association with the corticospinal tract.
And it gets input from the cortex, too.
Yes, it gets fibers directly from the primary motor cortex via the corticorubral tract, and it also receives branching fibers directly from the corticospinal tract itself as it passes through the midbrain.
So it gets cortical info two ways.
Essentially, yes. These fibers mostly synapse in the lower part of the red nucleus, known as the magnocellular portion. This part contains large neurons, remarkably similar in size to those Betz cells in the primary motor cortex.
And these large neurons in the red nucleus are the origin of the rubrospinal tract. Rubro meaning red.
Correct. The rubrospinal tract also crosses over to the opposite side in the lower brainstem, following a path immediately adjacent and slightly in front of the corticospinal tract in the lateral columns of the spinal cord.
And like the corticospinal tract, it terminates mainly on interneurons down in the spinal cord.
Yes, mostly on interneurons in the intermediate gray matter. But the sources note that some fibers do terminate directly on anterior motor neurons, similar to the corticospinal tract.
Okay. And it also connects with the cerebellum.
Yes, the red nucleus has close connections with the cerebellum, mirroring the strong motor cortex cerebellum links we already mentioned.
So this whole corticorubrospinal system essentially serves as an accessory route for motor commands. What's the functional difference? Why have two pathways doing similar things?
Well, the sources describe it as transmitting relatively discrete signals from the motor cortex. While the magnocellular red nucleus has a body map, it's generally less detailed than the primary motor cortex map, particularly in humans, where the red nucleus is relatively smaller compared to our massive cortex. Stimulation can cause contraction of a single muscle or maybe a small group.
And the key insight here is what happens if the main corticospinal pathway is damaged, like in a stroke.
This is where the rubrospinal tract's importance becomes really clear. If the corticospinal fibers are destroyed, discrete movements can still occur via this alternative corticorubrospinal pathway.
So it's a backup system.
Partially. However, the sources emphasize that the fine control of the fingers and hands is significantly impaired. Interestingly, wrist movements can often still be functional in this scenario, suggesting the rubrospinal tract contributes more significantly perhaps to proximal limb control or less refined movements.
So you might be able to move your wrist, but not your fingers precisely.
Exactly. But if both the corticospinal and rubrospinal pathways are blocked, then even those wrist movements are lost.
That really clarifies the specialization. Corticospinal seems absolutely essential for that fine finger dexterity, while rubrospinal is important for other discrete movements, maybe more in the wrist and forearm, and can serve as a partial backup.
It's a complementary system. And together, these two tracts, the corticospinal and rubrospinal, are referred to as the lateral motor system of the spinal cord.
Lateral motor system. Why lateral?
Because they travel in the lateral side columns of the spinal cord and primarily terminate on neurons controlling the more distal muscles of the limbs, those further away from the body's core, like hands and feet.
And this contrasts with?
This contrasts with the medial motor system, which includes tracts like the vestibulospinal and reticulospinal tracts that run more centrally medially in the cord and tend to control the axial muscles, the trunk and core muscles involved in posture.
Okay, lateral for limbs, medial for core. Got it. So we have the signals coming down these complex pathways. How do they actually trigger activity in the spinal cord motor centers to make muscles contract?
Right. The source details how the motor cortex's output is organized at a finer level. Like other cortical areas, the neurons in the motor cortex are arranged in vertical columns, fractions of a millimeter across, each containing thousands of neurons.
And each column acts like a processing unit, a functional module.
Yes, functioning as a unit to typically stimulate a group of synergistic muscles involved in a specific movement, or sometimes a single muscle. Within these columns, the pyramidal output cells are in layer 5, input signals arrive in layers 2, 4, and layer 6 connects to other cortical regions.
This columnar structure seems built for complex integration, doesn't it?
It is. The sources describe each column as an integrative system, combining multiple inputs to determine the final output signal. They can also amplify a signal, exciting many pyramidal fibers for the same or related muscles simultaneously.
Why is amplification needed?
Well, this amplification is necessary because stimulating just one pyramidal cell rarely causes a muscle contraction. You typically need 50 to 100 firing together, or in rapid succession, to get a definitive muscular response.
So one neuron shouting isn't enough, you need a chorus.
Pretty much. And this is where the concept of dynamic and static signals comes in. It's really cool.
Okay, tell me about dynamic and static signals.
This is a key insight into how the brain controls the force and timing of muscle contractions. The sources explain that each motor cortex column excites two distinct populations of pyramidal neurons that project down.
Two types of output neurons from the same column?
Yep, dynamic and static neurons. The dynamic neurons fire at a very high rate, but only for a short period, specifically at the beginning of a muscle contraction.
The burst at the start.
Right. Their role is to cause the initial rapid surge of force needed to start the movement quickly, get things moving. The static neurons, on the other hand, fire at a much slower rate, but they continue firing throughout the duration of the contraction.
So they maintain the force.
Exactly. Their role is to maintain the force of contraction for as long as needed. Dynamic for the burst, static for the sustained effort.
That's elegant.
Isn't it? The sources also point out that the red nucleus has a relatively greater proportion of dynamic neurons compared to the primary motor cortex, which has more static neurons.
Interesting. Any idea why?
They connect this back to the cerebellum's known role in initiating rapid movements. Maybe the red nucleus, strongly influenced by the cerebellum, is more involved in the get-going fast part. It's another hint at how these systems specialize and cooperate.
How important is sensory feedback from the muscles and limbs during the movement? Does the cortex get updates?
Absolutely vital for precision and control. It's not just fire and forget. When the motor cortex sends a command to contract a muscle, sensory signals race back from that activated body part, returning all the way up to the motor cortex neurons that initiated the command.
Real-time updates. What kind of feedback is this?
It comes from muscle spindles, which sense muscle length and how fast it's stretching, muscle tendon organs, which sense muscle tension or force, and tactile receptors in the skin over the muscles, sensing pressure and contact.
And this feedback can actually enhance the contraction. That seems like it could lead to runaway signals. How does that work?
It does sound a bit like positive feedback, but it's tightly controlled. The source describes this as positive feedback enhancement. Take the muscle spindles. They have their own tiny muscle fibers inside them, controlled by different neurons, physiomotor fibers.
Okay.
If these spindle fibers contract more than the main skeletal muscle fibers, the central part of the spindle stretches, sending signals back to the motor cortex saying essentially, hey, the main muscle isn't pulling hard enough to match the desired length.
The spindle is like a little target length sensor.
Exactly. The motor cortex pyramidal cells respond by firing more, further exciting the main muscle fibers, helping them catch up to the spindle contraction and achieve the intended movement accurately.
That's a clever built-in system to ensure the desired contraction level is actually met, like a servo mechanism.
It really is. Or think about tactile receptors. When you grasp something, the pressure on the skin provides feedback. This feedback can loop back to the motor cortex and cause further excitation, increasing grip strength if the object starts to slip, for instance. It's a continuous, self-adjusting control loop.
Okay, so how do these signals from the cortex and red nucleus finally connect with the motor neurons in the spinal cord to make the muscle fire? We know they mostly hit interneurons, but what about that direct connection?
Right, let's look at figure 56-6 in the source, showing the spinal cord cross-section. The corticospinal and rubrospinal tracts run in the dorsal parts of the lateral white columns, and as we said, mostly terminate on interneurons in the intermediate gray matter.
But you mentioned a key exception.
Yes, and this is a critical detail for understanding fine motor control, especially in humans. In the cervical enlargement of the spinal cord, the part that controls the hands and fingers, large numbers of both corticospinal and rubrospinal fibers terminate directly on the anterior motor neurons.
Directly onto the final output neurons, bypassing the interneuron.
Exactly. This direct connection allows for the extremely rapid and precise control of individual or small groups of muscles in the hands and fingers, the hallmark of human dexterity. It directly correlates with the huge representation of hands in the motor cortex and the presence of those superfast Betz cells. It's the express lane for fine control.
That makes perfect sense. And the spinal cord itself isn't just a passive receiver of these commands, is it? It has its own pre-programmed patterns.
Crucially, yes. The sources remind us that the spinal cord circuitry itself provides many basic reflex patterns, like the stretch reflex we just touched on, the withdrawal reflex, pulling away from pain, and reciprocal innervation.
Reciprocal innervation? What's that again?
That's the mechanism where activating one muscle group, like your biceps to bend your elbow, automatically causes inhibition or relaxation of the opposing muscle group, your triceps.
Ah, right, so you don't fight yourself.
Exactly. When the brain sends a command signal down to the spinal cord motor neurons, it doesn't have to code every single detail from scratch. It activates and utilizes these existing spinal cord patterns.
So the brain tells the cord what goal to achieve, and the cord's built-in circuitry handles some of the details of how to execute it smoothly.
That's a great way to put it. The stretch reflex, for instance, is always active and helps to dampen oscillations during movements initiated by the brain, making them smoother, less jerky. Reciprocal innervation is also constantly present. When the brain excites an agonist muscle, the spinal cord circuitry automatically handles inhibiting the antagonist, ensuring smooth movement without needing a separate relax antagonist command from the brain.
And what about more complex patterns like walking or scratching? Does the brain trigger those in the cord too?
Yes, the sources state that other reflex mechanisms in the cord, like those for stepping walking, central pattern generators, scratching, and various postural mechanisms, can be initiated by relatively simple command signals from the brain. A high-level command like walk, sent from the brain, can activate the complex stepping pattern generator within the spinal cord.
So the brain says walk, and the spinal cord runs the walking program. Essentially, yes. This means many fundamental motor activities can be executed using these built-in patterns, with the higher centers just providing the command and modulation.
Okay, now let's shift gears slightly and talk about what happens when this intricate system is damaged. The sources discuss lesions, particularly those caused by stroke.
Right. Stroke, caused by a ruptured blood vessel causing a hemorrhage or a blood clot blocking an artery, thrombosis, is a very common way damage occurs. It often affects the motor cortex or the corticospinal tract where it passes through that narrow internal capsule we mentioned.
What's the effect of removing just the primary motor cortex, the area specifically containing those Betz cells? Let's say in a very precise surgical removal.
The sources describe the outcome. It results in varying degrees of paralysis of the muscles on the opposite side of the body. However, and this is important, if the adjacent areas like the premotor, supplementary motor, and crucially, the basal ganglia are not damaged, gross postural movements and the ability to fixate a limb in a position can still occur.
But you lose the fine control,
Okay.
the dexterity. Precisely. There's a profound loss of voluntary control of discrete movements of the distal limbs, particularly the hands and fingers. The ability of the muscle itself to contract isn't lost, but the ability to initiate and precisely control those fine movements is gone.
So the primary motor cortex is absolutely essential for the voluntary initiation of finely controlled movements, especially for the hands and fingers.
That's the conclusion from those observations, yes. It confirms its role in the fingernail details of movement.
Okay, but what about the muscle spasticity often seen after a stroke? That seems different from just a loss of fine control. It's like there's too much muscle tone. Muscles are stiff and resist movement.
This is a key distinction the sources make and a critical point of insight. It's often misunderstood. While removing just the primary motor cortex's constant tonic influence might initially cause some reduced tone, hypotonia.
Muscles might be a bit floppy at first.
Briefly perhaps, but typical stroke lesions are broader. They often involve the primary motor cortex and adjacent parts of the brain, critically including those accessory motor pathways that travel near the cortex, often through the internal capsule, where strokes commonly occur.
And damaging these adjacent pathways, these accessory pathways, is what causes the spasticity.
Almost invariably, yes. Stroke lesions involving these accessory pathways lead to muscle spasticity, that's severe involuntary muscle stiffness on the opposite side of the body due to the pathways crossing over.
Okay, so what's the mechanism? Why does damaging other pathways cause stiffness?
The critical insight from the sources is that the main cause of this spasticity isn't the primary motor cortex damage itself, but damage to those accessory pathways, specifically the ones originating from the non-pyramidal motor cortex areas.
And what do those accessory pathways normally do?
They normally exert a continual inhibitory effect on certain brainstem motor nuclei, specifically the vestibular and reticular nuclei we'll discuss next. These brainstem nuclei are naturally excitatory and help support antigravity muscles.
So these accessory pathways from the cortex normally keep the brainstem support systems in check. Tell them to calm down a bit.
Exactly. They inhibit them. When these inhibitory pathways are damaged by the stroke, the brainstem nuclei are disinhibited or released from that cortical control. They become spontaneously and excessively active.
And that causes the spastic muscles.
Yes. This leads to the profound, excessive spastic tone in the antigravity muscles that is so characteristic of stroke recovery. It's not the cortex overstimulating the muscles, but the cortex's inhibitory control over lower brainstem centers being lost, allowing those lower centers to become overactive.
Wow. That's a completely different way of thinking about spasticity. It's a loss of inhibition, not excessive excitation from the damaged area itself.
That's the mechanism described in the sources, and it's a vital insight into the pathology. It highlights the crucial balance of excitation and inhibition needed for normal tone and movement, orchestrated across different brain levels.
This leads us perfectly into discussing the critical role of the brainstem in motor function, particularly in maintaining that fundamental support against gravity and posture.
Absolutely. The brainstem, comprising the medulla, pons, and midbrain, or mesencephalon, is far more than just a simple relay station for signals passing through.
It has its own jobs to do.
Definitely. While it does extend spinal cord functions upwards, containing motor and sensory nuclei for the face and head, similar to how the spinal cord handles the neck down, it also contains many vital centers for special control functions.
What kinds of special functions?
The sources list key ones: control of respiration, the cardiovascular system, parts of the digestive system, many stereotype body movements, equilibrium, and eye movements. It's a busy place.
And for our focus on movement and equilibrium.
For that, the reticular nuclei and the vestibular nuclei within the brainstem are particularly important. They play a central role in controlling whole body movement and maintaining balance.
Okay, let's look at figure 56-7 showing these areas. The reticular nuclei seem spread out.
They are. They're broadly organized into two major groups with basically antagonistic functions.
Pontine and medullary reticular nuclei.
Right. The pontine reticular nuclei are located mainly in the pons and extend up into the midbrain. Their primary role is to excite the anti-gravity muscles.
The muscles that keep you standing up.
Exactly. The medullary reticular nuclei run throughout the medulla, more towards the front and midline. Their primary role is to relax or inhibit those same anti-gravity muscles.
So one group turns on the stand-up straight muscles, the other group turns them off. An accelerator and a brake for posture.
That's a good analogy. The pontine reticular system sends excitatory signals downward via the pontine reticulospinal tract, which travels in the anterior front column of the spinal cord, as shown in figure 56-8.
Where do they connect?
These fibers terminate on the medial anterior motor neurons, the ones controlling the axial muscles of the vertebral column and the extensor muscles of the limbs, precisely the muscles needed to support the body against gravity.
And this pontine system is naturally quite active.
Yes. The sources state it has a high baseline level of natural excitability. Plus, it receives strong excitatory input from the vestibular nuclei, which sense balance, and the deep nuclei of the cerebellum.
What happens if it's left unchecked?
The sources give a powerful illustration. If this pontine excitatory system is left unopposed by inhibition, it can cause such powerful excitation that a four-legged animal can stand rigidly and support itself against gravity without any input from higher brain centers at all.
Wow, just the brainstem is enough to hold an animal up.
It shows its fundamental, almost automatic support function.
Okay, counterbalancing this powerful excitatory system is the medullary reticular system, the inhibitory one.
Right. This system sends inhibitory signals via the medullary reticulospinal tract, which travels in the lateral column of the spinal cord, figure 56-8 note, a different location than the pontine tract, but targeting the same anti-gravity motor neurons.
And what provides input to this inhibitory system? What tells it to hit the brakes?
It receives strong input from the cerebral cortex via the corticospinal tract, the red nuclei via the rubrospinal tract, and other motor pathways, including the basal ganglia. These higher centers normally activate the medullary inhibitory system.
Why?
This activation serves to counterbalance the natural excitability of the pontine system. It prevents your muscles from being abnormally tense all the time and allows for controlled relaxation and movement.
So higher brain areas can choose to activate the pontine system more for strong support, or activate the medullary system to relax muscles needed for other actions. It's adjustable.
Precisely. This excitatory-inhibitory balance, controlled by signals from the cortex, basal ganglia, cerebellum, and other areas, is the brainstem's mechanism for providing the necessary background muscle contractions for maintaining posture and standing, while also allowing specific muscles to relax when needed for other activities. It's a controllable gain system.
The vestibular nuclei also provide excitatory support for anti-gravity muscles, working closely with the pontine system you mentioned.
Yes, they function in strong association. The vestibular nuclei send powerful excitatory signals down the lateral and medial vestibulospinal tracts, also in the anterior columns of the spinal cord, figure 56-8.
How important is their contribution?
The sources note that without the support from the vestibular nuclei, the pontine system loses a significant amount of its excitatory effect, especially on the axial muscles of the trunk.
And their specific role is linked to equilibrium, right? Not just general excitation.
Exactly. The vestibular nuclei selectively control those excitatory signals to different anti-gravity muscles based on input they receive from the vestibular apparatus in the inner ear. This selective control is vital for maintaining equilibrium, constantly adjusting posture in response to perceived changes in balance. We'll dive into that vestibular system next.
Okay, but first, the sources use the example of decerebrate rigidity to dramatically illustrate the importance of this brainstem balance between excitation and inhibition. What is that?
This is a condition observed experimentally, or sometimes after severe brain injury. It occurs when the brainstem is sectioned below the level of the midbrain, but leaving the pontine, medullary, and vestibular systems intact and connected to the spinal cord.
So you cut off the inputs from the cortex, basal ganglia, and red nucleus.
Essentially, yes. You remove those higher-level modulatory inputs.
And the result is severe spasticity, like we discussed with stroke.
Yes, but in a very specific pattern. A profound, rigid spasticity develops specifically in the anti-gravity muscles, the extensors of the limbs, trunk, and neck. It doesn't affect all muscle groups equally.
And the cause is that loss of inhibition from higher centers.
Precisely. The sectioning blocks the normal, strong inhibitory input that the medullary reticular nuclei receive from the cerebral cortex, red nuclei, and basal ganglia. Without this critical inhibition, the medullary inhibitory system becomes essentially non-functional.
So the brakes are off?
Completely. This allows the pontine excitatory system, along with the vestibular excitatory input, to become completely overactive and unopposed, resulting in the severe rigidity of the antigravity muscles. It's a clear demonstration of how higher center inhibition is absolutely necessary to temper the brainstem's inherent excitatory drive for posture.
So the brainstem provides this fundamental, powerful support system, and higher centers modulate that system, turning the gain up or down, inhibiting or exciting to allow for controlled movement and relaxation.
That's the core concept. And damage to the modulatory inputs from above leaves the fundamental support system running unchecked, leading to rigidity or spasticity.
Okay, this leads perfectly into how we actually sense our orientation and movement in space, the vestibular sensations, and how they contribute to maintaining equilibrium. Where does the system live?
This system begins with the vestibular apparatus, our sensory organ for detecting equilibrium. It's located within the inner ear, right next to the cochlea, the hearing organ. Figure 56-9 shows its structure. It's housed within the bony labyrinth in the temporal bone and contains the functional part, the membranous labyrinth.
Which includes the cochlea for hearing, but also the structure's important for balance, you said.
Yes, specifically the 3 semicircular canals and 2 larger chambers called the utricle and the saccule. These latter 3 are the integral components of the equilibrium mechanism.
Let's start with the utricle and saccule. What do they detect? What's their job?
The sources explain they detect the orientation of the head with respect to gravity, essentially, whether your head is upright, tilted, or lying down. They sense static position and also linear acceleration.
How do they do that?
Each chamber has a small sensory area on its inner surface called a macula. Think of it like a little patch of sensory cells.
And they're oriented differently within the utricle and saccule.
Yes, and that's key. The utricle macula is mainly horizontal, making it most sensitive to head orientation when you're upright, detecting tilt side to side or front to back. The saccule macula is mainly vertical, playing a larger role when you're lying down, or sensing up-down movements like in an elevator.
And these macula contain the actual sensory hair cells and those little crystals, the statoconia.
Correct. Each macula is covered by a gelatinous layer. Embedded within this layer are thousands of hair cells, the actual sensory receptors. Projecting from the surface of this gelatinous layer are tiny calcium carbonate crystals called statoconia, sometimes called otoliths.
Statoconia balance stones.
Pretty much. The cilia, the little hair-like projections from the top of the hair cells, project up into this gelatinous layer and are surrounded by the statoconia.
And the statoconia are key because of their weight, right? They're heavier than the surrounding stuff.
Precisely. They have a specific gravity 2 to 3 times greater than the surrounding fluid and tissue. So the weight of the statoconia pulls on the gelatinous layer and bends the cilia of the hair cells in the direction of gravitational force. When you tilt your head, gravity pulls the heavy statoconia, which shifts the gel, which bends the hairs.
Okay, so the hairs bend. How does that bending get converted into a neural signal? The sources describe a fascinating directional sensitivity based on something called the kinocilium.
This mechanism is quite elegant, yeah. As shown in figure 5610, each hair cell has maybe 50 to 100 smaller hair-like projections called stereocilia, and one single larger cilium called the kinocilium, always located off to one side of the bundle.
One big one, lots of small ones.
Right. And there are minute filamentous attachments, like tiny threads, connecting the tip of each stereocilium to the next taller one beside it, ultimately linking the whole bundle to the kinocilium.
So they're all physically linked together, like a tiny, springy bundle.
Exactly. When the statoconia's weight causes the gel to shift and bends the bundle of cilia towards the tall kinocilium, it increases tension on those filamentous attachments. This tugs on the stereocilia, pulling them slightly outward and physically opening hundreds of ion channels, specifically cation channels, near their bases.
Opens gates for ions to flow in.
Right. Positive ions, mainly potassium, from the surrounding fluid called endolymph, pour into the hair cell, causing the cell membrane to depolarize it, becomes electrically excited.
And bending the other way, away from the kinocilium.
Bending the cilia away from the kinocilium reduces the tension on those attachments, allowing the ion channels to close. This reduces the inflow of positive ions and causes the cell membrane to hyperpolarize it, becomes inhibited or less excitable.
So bending one way excites the hair cell, bending the other way inhibits it. It's directionally sensitive.
Exactly. And the nerve fibers synapsing with these hair cells maintain a continuous baseline firing rate, maybe around 100 impulses per second, even when the head is perfectly still and upright.
They're always on to some degree.
Yes. Bending towards the kinocilium increases this firing rate significantly, maybe up to several hundred per second. Bending away decreases it, potentially stopping the firing completely for a moment.
Okay, but how does this detect head orientation? All the hair cells doing the same thing.
No, and this is the clever part. The hair cells in each macula are oriented in many different directions. Their kinecilia point different ways. So a specific head orientation relative to gravity will bend different sets of cilia in different ways, some towards their kinecilium, some away, some barely at all.
Ah, so it's the pattern across all the differently oriented cells.
Precisely. This creates a unique pattern of excitation and inhibition across the thousands of nerve fibers coming from the maculae for each possible head position. This distinct pattern is what the brainstem and cerebellum interpret to know, very precisely, the head's position relative to gravity.
And this system, using the maculae, is very effective for maintaining equilibrium when you're mostly upright.
Yes. The sources note it's incredibly sensitive, able to detect disequilibrium when leaning as little as half a degree. Tiny deviations create a detectable change in that pattern.
The maculae also detect linear acceleration, you mentioned. Like when you're in a car that suddenly speeds up or an elevator starts moving. How does that work?
It uses the same principle, but relies on inertia instead of just gravity. If your body is suddenly thrust forward, accelerates. The statoconia, having greater mass and inertia than the surrounding fluid, will tend to lag backward relative to the rest of the macula and the hair cells.
Because they're heavier, they resist the change in motion more.
Right. This backward lag bends the cilia as if you were tilting backward, sending signals indicating a sense of falling backward. Your nervous system then automatically triggers postural adjustments, usually causing you to lean forward slightly.
Why lean forward?
You lean forward until the forward shift of the statoconia, due to the lean, exactly counteracts the backward lag due to the acceleration. At that point, the cilia return to their neutral equilibrium position. The signals normalize, and your nervous system senses that you're back in balance relative to the combined forces of gravity and acceleration.
That's clever. But the maculae don't detect constant linear velocity, just the change in velocity, the acceleration or deceleration?
Correct. Once you reach a steady speed, the statoconia catch up, the relative lag disappears, and the signal returns to baseline. The sources use the example of a runner. The forward lean isn't due to the steady speed itself, but to the acceleration needed to reach that speed, or perhaps the constant force needed to counteract air resistance, which might be sensed partly by skin receptors, not just the maculae.
Okay, so maculae handle gravity, static position, and linear acceleration. What about the semicircular ducts? They detect something different. Head rotation or angular acceleration?
Yes, and their physical arrangement is key to how they do this. There are 3 semicircular ducts, anterior, posterior, and lateral or horizontal, in each inner ear. They are positioned at roughly right angles to each other, like the 3 sides meeting at the corner of a box.
So they represent all 3 planes in space, X, Y, and Z axes.
Essentially, yes. This allows them to detect rotation in any direction, nodding your head yes, pitch, shaking your head no, yeah, or tilting your head side to side, roll. The source notes their specific orientation relative to gravity when the head is tilted slightly forward, which is typical for human standing.
How do they work mechanically? They're filled with fluid, right?
Yes. Each duct is a continuous loop filled with a fluid called endolymph. At one end of each duct, there's an enlarged swelling called the ampulla. When your head begins to rotate in the plane of one of the ducts, the bony duct and the ampulla move with your skull.
But the fluid inside?
The endolymph inside tends to lag behind due to its inertia. It resists the change in motion. This creates a relative flow of endolymph within the duct, moving in the direction opposite to your head's rotation.
Like the water sloshing in a bucket when you start to turn it?
Exactly like that. And this fluid flow specifically affects a structure inside the ampulla.
What's in the ampulla?
There's a crest of tissue called the crista ampullaris, and sitting on top of this crest is a gelatinous, sail-like mass called the cupula. This cupula stretches across the ampulla like a swinging door.
Okay.
Embedded in the crista, with their cilia projecting up into the gelatinous cupula, are hundreds of hair cells, similar to those in the maculae, shown in figure 5611.
And the flowing endolymph pushes on this cupula?
Yes. The relative flow of endolymph pushes against the cupula, bending it like wind pushes a sail. And because the hair cell cilia are embedded in it, bending the cupula bends the cilia.
And these hair cells also have a kinecilium for direction sensing.
Yes, and crucially, all the kinecilia on the hair cells within a single ampulla are oriented in the same direction. This means that fluid flow And cupula bending in one direction will depolarize all the hair cells in that ampulla, while flow in the opposite direction will hyperpolarize them all.
So each canal gives a clear signal for rotation in its specific plane.
Exactly. Bending the cupula in the direction of the kinocilia excites the hair cells and increases nerve firing. Bending it the opposite way inhibits them and decreases firing. This sends signals via the vestibular nerve to the brain about the rate and direction of change of rotation, the angular acceleration in each of the three planes.
The discharge pattern from these hair cells during rotation is particularly revealing, isn't it? Figure 5612 shows this.
It is very informative. Like the maculae cells, these hair cells have a baseline tonic discharge rate when the head is still. When rotation suddenly begins in the plane of the canal, the endolymph lags, the cupula bends towards the kinocilia, depending on the canal and direction, and the hair cell discharge rate increases dramatically.
A big spike in activity.
Right. But importantly, this increased rate adapts over the next several seconds, even if the rotation continues at a constant velocity. The firing rate gradually returns towards the baseline tonic level.
Why does it adapt? Why does the signal fade if you keep rotating steadily?
It's because the endolymph, due to friction with the walls of the duct, eventually starts rotating at the same speed as the duct itself. Once the fluid catches up, there's no more relative flow, so the pressure on the cupula decreases. The elastic cupula then slowly returns to its neutral unbent position, and the hair cell firing goes back towards its resting rate.
So it mainly signals the start of rotation.
Exactly. The initial burst of activity signals the angular acceleration, the change from rest to rotation. Then the signal decays during constant velocity rotation.
And what happens when the rotation stops?
The opposite effect occurs.
Yeah.
Your head and the duct stop rotating, but the endolymph, now moving with the duct, keeps swirling for a moment due to its inertia.
Like the water sloshing the other way when you stop the bucket?
Precisely. This continuing fluid flow pushes the cupula in the opposite direction to the initial push. This bends the cilia away from the kinocilium, hyperpolarizing the hair cells and causing their discharge rate to stop entirely or drop well below the baseline for a few seconds.
So silence or near silence signals stopping.
Yes. Then, as the endolymph stops swirling and the cupula returns to its neutral position from the opposite side, the discharge gradually returns to its normal tonic rate. So the semicircular ducts give a signal of one polarity, increased firing, when rotation begins, and the opposite polarity, decrease or stop firing when it stops. They are exquisitely sensitive to changes in rotation.
This leads to the really critical insight, the source called the predictive function of the semicircular duct system. This sounds important.
This is perhaps the most advanced aspect of the vestibular system's contribution to balance. The key is that the semicircular ducts detect the beginning or stopping of rotation. They detect angular acceleration, not steady rotation or static imbalance.
Whereas the maculae detect imbalance after it has already occurred, right? They sense the tilt itself.
Right, precisely. So if you suddenly turn while running or walking, the maculae won't signal that you're off balance until you're already starting to tilt or fall. That might be too late for a quick correction.
But the semicircular ducts detect the turn itself as it's happening?
Yes. They detect the turning motion, the angular acceleration, before you've fully lost your balance. This information serves as an anticipatory signal to the brain. It tells the brain that disequilibrium is likely about to happen because you've just initiated a turn.
Allowing the brain to make adjustments before you actually fall. Proactive balance control.
Exactly. This allows the equilibrium centers in the brainstem and cerebellum to receive this early warning and make anticipatory, preventive adjustments to your posture and muscle activity. You can shift your weight, adjust your stance, maybe tense certain muscles to maintain balance before the actual disequilibrium develops significantly. It's a predictive control system, helping you stay upright during dynamic movements.
That's amazing. It's like having a little predictive gyroscope in your head. And the sources link this predictive function strongly to the cerebellum, don't they?
They do, very strongly. The flocculonodular lobes of the cerebellum, in particular, are intimately connected with the semicircular ducts and are especially concerned with processing these dynamic equilibrium signals related to rotation and changes in motion.
What happens if those lobes are damaged?
Destruction of these lobes in animals causes symptoms that almost perfectly mirror the destruction of the semicircular ducts themselves. A severe loss of dynamic equilibrium during rapid changes in direction or speed, but interestingly, less disturbance when the animal is just trying to maintain a static posture.
So the cerebellum, especially these lobes, is using the semicircular duct information for this predictive balancing act during movement.
Yes. The cerebellum is widely seen as a key predictive organ in motor control, using incoming sensory information to anticipate the results of movements and aid in rapid, smooth adjustments. The sources also mention the uvula, another part of the cerebellum, as having an important role in static equilibrium, perhaps more linked to the maculae input.
Beyond just maintaining posture and balance, the vestibular system is absolutely vital for stabilizing our vision during movement, isn't it? The vestibulo-ocular reflex.
Absolutely essential.
Yeah.
Imagine trying to read a sign while walking. If your eyes just bounced around with every step, the world would be a blur. To get clear images, your eyes need to be able to fixate on objects, and that image needs to be held relatively stable on the retina, despite head movements.
How does the vestibular system make that happen?
Every time your head rotates, even slightly, the semicircular ducts detect that motion and send signals that trigger powerful, very fast reflexes. These signals travel via the vestibular nuclei and a critical pathway called the medial longitudinal fasciculus, directly to the motor nuclei that control your eye muscles, the oculomotor, trochlear, and abducens nuclei.
And the result is the eyes move in the opposite direction of the head turn automatically.
Yes, precisely and automatically. The reflex causes your eyes to rotate in a direction and with a velocity that is almost perfectly equal and opposite to your head's rotation. This keeps your gaze fixed on a specific point in the world, effectively canceling out the head movement and allowing you to maintain a stable visual field. The sources reference this reflex, noting its detailed description elsewhere, highlighting just how important it is for functional vision during everyday life.
So a rapid direct reflex arc from the inner ear rotation sensors to the eye muscles keeps the world from blurring when we move our heads. That's critical.
Incredibly critical.
Okay, so the vestibular apparatus is providing all this crucial information about gravity, acceleration, and rotation. But it's not the only source of input for maintaining equilibrium, right? Other factors contribute significantly.
That's absolutely right. Balance is truly a multisensory process. One vital set of additional inputs comes from the proprioceptors in your neck.
Proprioceptors, those are the sensors in muscles and joints telling you limb position.
Exactly. The vestibular apparatus only tells you about the orientation of your head in space relative to gravity or rotation. But your brain also needs to know the orientation of your head with respect to your body. Are you tilting your head, or is your whole body tilting?
Ah, that's a key distinction. And that information comes from sensors in the neck.
Yes, primarily from proprioceptor sensors detecting stretch and position in the muscles and joints of your neck. This information is transmitted directly to the vestibular and reticular nuclei in the brainstem, and also indirectly via the cerebellum. The joint receptors in the neck are particularly important for this head-on body information.
How do the neck signals interact with the vestibular signals? Do they work together or against each other?
The sources explain a key interaction that helps the brain differentiate head tilt from body tilt. If you bend only your neck, tilting your head relative to your stationary body, the impulses from the neck proprioceptors signal that head-on body movement. These signals actually oppose the signals coming from the vestibular apparatus, which are signaling head tilt relative to gravity.
Why oppose?
Because your body's orientation relative to gravity hasn't changed, only your head's relative to your body. This opposition, this conflict between neck and vestibular signals prevents you from feeling a sense of disequilibrium or dizziness just from tilting your head. The brain interprets this conflict as head movement relative to body rather than whole body tilting.
But if you lean your entire body over, head and body together,
Then the vestibular signals, indicating a change in orientation relative to gravity, are not opposed by the neck proprioceptors, because the head hasn't moved significantly relative to the neck body. So in this case, the brain does perceive a change in the equilibrium status of your entire body, and you feel like you're leaning.
that's a very clever way to disambiguate those signals. What about other proprioceptive and exteroceptive information?
Yes, those contribute too. Signals from pressure receptors in the soles of your feet, for example, inform your brain about how your weight is distributed. Is it equal between your feet? More on your heels or toes. This is crucial for subtle postural adjustments.
And exteroceptive, from outside the body.
Right. The source mentions things like the feeling of air pressure on the front of your body when running, which provides input about external forces that need to be counteracted, prompting postural adjustments like leaning forward into the wind or resistance.
And let's not forget vision. It seems like a major backup system for equilibrium.
It's incredibly important, especially if other systems like the vestibular or proprioceptive systems are compromised. Even if someone has lost their vestibular function completely, they can often maintain near normal equilibrium using visual information alone. especially when moving slowly.
How does vision help with balance specifically?
Even very slight movements or sways of your body cause instantaneous shifts in the images falling on your retina. Your visual system detects this optic flow or image shift very rapidly. This visual information about body sway is processed and sent to the equilibrium centers in the brain, allowing for corrective postural adjustments.
So you use visual cues to tell if you're moving when you shouldn't be.
Exactly. The sources note that people with bilateral vestibular destruction, who might be profoundly unbalanced and fall easily in the dark or with their eyes closed, can often regain near normal balance when their eyes are open, at least during slow, deliberate movements. Rapid movements still cause significant difficulty because vision isn't as good as the vestibular system at detecting very fast accelerations or providing that predictive signal.
So, maintaining balance is a truly multisensory feat, constantly integrating information from gravity sensors, maculae, rotation sensors, semicircular ducts, body position sensors, proprioceptors in neck, feet, etc., and vision.
It's a continuous stream of data from multiple channels, all feeding into the brainstem and cerebellum to inform a complex, coordinated, and largely unconscious motor response to keep you upright.
Okay, let's look briefly at the main neuronal connections of the vestibular apparatus with the central nervous system. Where do those signals go first?
The vestibular nerve, which carries the signals from the hair cells in the maculae and ampullae, sends the vast majority of its fibers directly into the brainstem, terminating in the vestibular nuclei. These nuclei are located at the junction of the medulla and the pons.
Do all fibers end there?
Most do, but not all. Some fibers bypass the vestibular nuclei and go directly to the brainstem reticular nuclei, those posture control centers we discussed. And importantly, some fibers go directly to specific nuclei within the cerebellum, particularly the vestigial nuclei and parts of the cerebellar cortex called the uvula and the flocculonodular lobes.
So direct lines to the cerebellum bypassing even the first brainstem relay. That emphasizes the cerebellum's importance.
Absolutely. It gets privileged, rapid access to vestibular information.
And these second-order neurons, the ones starting from the vestibular nuclei, where do they send signals?
They distribute signals widely. They send strong projections to the cerebellum, reinforcing that connection. They send signals down via the vestibulospinal tracts to the spinal cord motor neurons, influencing posture. They send signals up and down via medial longitudinal fasciculus, which is crucial for coordinating eye, head, and neck movements. And they send signals to other brainstem areas, particularly the reticular nuclei, further integrating balance and posture control.
So the primary reflex pathway for maintaining equilibrium looks roughly like vestibular nerves, vestibular nuclei and our cerebellum, reticular nuclei and ascending tracts, vestibulospinal, reticulospinal, to the spinal cord motor neurons controlling anti-gravity muscles.
Correct. This pathway forms the core circuitry that constantly modulates the balance of excitation and inhibition in the anti-gravity muscles of the spinal cord, providing that automatic subconscious equilibrium control.
And the cerebellum's specific roles tie back to those dynamic and static functions we discussed.
Exactly. The sources re-emphasize that the flocculonodular lobes are especially linked to the dynamic equilibrium signals coming from the semicircular ducts. Damage here messes up balance during movement and rapid changes. The uvula, another cerebellar part, seems more involved in static equilibrium, likely processing information from the maculae about head position relative to gravity.
What about signals going up from the vestibular system to our conscious brain? Do we get conscious awareness of balance?
Yes, we do. From the vestibular nuclei and the cerebellum, signals ascend. Some go via the medial longitudinal fasciculus to the eye muscle nuclei for those vital corrective eye movements, the vestibulo-ocular reflex. Other pathways carry signals further up, eventually reaching a primary cortical center for equilibrium.
A balance cortex, where is this located?
The sources pinpoint it, based on functional studies, likely in the parietal lobe, deep within the folds of the sylvian fissure. Interestingly, it seems to be located near, but distinct from, the auditory cortex area, which is on the superior temporal gyrus.
And the function of this cortical area?
These ascending signals are what apprise the psyche, as the source puts it, of a body's equilibrium status. They give rise to our conscious perception of balance, motion, and orientation in space. It's how you know if you're upright or tilted or spinning.
Okay. Finally, before we wrap up, the source touches on a really fundamental point: the role of brainstem nuclei in controlling subconscious, stereotyped movements, even completely without input from the higher cortex.
This is powerfully illustrated by the tragic but informative example of babies born with anencephaly, a severe birth defect, where they lack brain structures above the level of the mesencephalon, midbrain. They're missing their entire cerebral cortex, basal ganglia, thalamus, etc.
So essentially just having a brainstem and spinal cord, what can they do?
The sources describe that despite missing almost their entire forebrain, these infants can still perform a surprising array of complex, stereotyped movements.
Like what kind of movements?
They can often suckle, extrude unpleasant food from their mouths, move their hands towards their mouths, yawn, stretch, cry, sometimes follow objects with their eyes and head, and even show some basic postural responses, like attempting to pull themselves to a sitting position if pressure is applied to their legs.
That's absolutely remarkable. It demonstrates that the core circuitry for many fundamental, seemingly complex, stereotyped motor functions is actually integrated right there at the brainstem level.
It really does. It shows that there's a foundational layer of quite sophisticated motor control that exists entirely below the level of conscious cortical control. The brainstem isn't just a simple relay. It contains its own complex, built-in motor programs that are essential for basic survival functions and interactions, ready to run even without cortical input.
Wow. We have covered a lot of ground here, really gone deep into the mechanics. To try and summarize this journey, aiming for that mastery level understanding, we started with the motor cortex, the planning center for voluntary movement.
Right, seeing how it works with the premotor and supplementary areas for planning and postural setup.
And sends signals down those two main routes, the direct corticospinal tract, especially for fine control via Betz cells and direct connections.
And the accessory corticospinal pathway, both using constant sensory feedback for fine-tuning.
And we saw how these descending pathways don't just dictate, they activate and utilize the pre-programmed patterns within the spinal cord itself, like reciprocal inhibition.
Then we explored the absolutely critical background control provided by the brainstem, its reticular nuclei, providing that adjustable excitatory, pontine, and inhibitory medullary support for anti-gravity posture, heavily influenced by signals from the vestibular nuclei and modulated by higher centers.
And we saw how damage interrupting that higher control, that inhibition, leads to things like spasticity or decerebrate rigidity because the brainstem systems are released.
Exactly. Then we delved into the incredible sensory side of balance with the vestibular apparatus, the utricle and saccule, with their statoconia detecting gravity and linear acceleration through patterned hair cell activity.
And those amazing semicircular ducts with the endolymph and cucula detecting rotation, crucially providing that predictive signal to the cerebellum to anticipate imbalance during movement.
Plus the vital role of the vestibulo-ocular reflex for stabilizing vision and the integration of all that with input from neck proprioceptors, body sensors, and vision itself.
It's truly a symphony of different systems: cortical planning, subcortical modulation from basal ganglia and cerebellum, brainstem foundational support and reflexes, spinal cord pattern execution, and exquisite multimodal sensory feedback, all communicating constantly.
The conscious command from the cortex is just the starting gun for this incredibly complex race involving modulation, background support, pattern generation, prediction, and real-time adjustment happening at multiple levels simultaneously. It all has to work together seamlessly.
And understanding how these components work and how disruptions like stroke impact that delicate balance of excitation and inhibition, particularly the release of those powerful brainstem systems, really elevates our understanding beyond just muscle weakness. It reveals the hidden brilliance, the intricate machinery behind seemingly simple acts like standing still or reaching for something.
It definitely makes you appreciate the incredible complexity and constant computation required just to remain upright and navigate the world without consciously thinking about every tiny adjustment.
Which leaves us with this final thought to consider. We've spent this time detailing millions upon millions of neurons, thousands of specialized fiber tracts, signals firing hundreds of times every second, intricate feedback loops, predictive calculations based on fluid dynamics in your inner ear, all happening right now, far, far below your conscious thought, just so you can maintain your posture, perhaps hold a glass, maybe walk across the room and listen to this. How much of what you experience as you and your seemingly effortless ability to interact with the world is actually built upon this breathtakingly complex, constantly running, automatic biological machine operating entirely in the background?
That is a truly profound thought on which to end our deep dive. Makes you reconsider what control really means.
Indeed. Thank you for joining us on this deep dive into the neural control of movement and equilibrium.